All right, I think we can get started. Sorry, let me use a different mic. Okay. Well, welcome everybody, and thank you all for showing up to this another session of our meeting on molecular ML. Today we're going to be talking about this really exciting paper coming from the Baker Lab about RF diffusion, and I'm sure many of you have heard of this, or at least the network that it was built on, RoseTTAFold. But we'll be getting into the specifics of this network as well as how it was trained and some of the extensions that have been applied to it. But before we do that, like usual, we're going to start with just some logistics. Once again, we're going to meet every 2 weeks. So the next meeting is going to be in 2 weeks from today, which is October 25th. For that meeting, I'm actually going to be unavailable. So this opens up the opportunity for anybody else who's willing to or interested in presenting a paper or talking about some specific work related to what we're discussing. If you're interested, please reach out to me. Otherwise, we'll figure something out, reschedule. So please reach out to me if you're interested.
So the atom-based RF diffusion model came out yesterday. That might be a good one to talk about in 2 weeks. What do we think? There's also the one around pocket flow. It's similar group as the RF diffusion or as the diff-doc group. Each of these would be fun to do. So I think I'll be here. I'm happy to help moderate if other people would want to talk about one or both of those.
Awesome. Yeah.
Okay.
So
we'll
please reach out to
us
us
on the Slack.
Yeah, we'll figure something out. Cool. So like usual, there's a Google form to select the papers. This isn't really relevant for this next meeting since it's going to be a little different, but we'll probably just talk about either all atom diffusion or the pocket stuff like Matt just suggested. But a little different from usual, we're actually going to separate the papers into some more foundational versus application-based papers. That way, if people are interested in learning more about the foundations of these models, we can dive into that and kind of build up the theory as well. Cool. So today we're talking about RF diffusion. And this is really a nice bridge between last session because we talked about frame diff. And these are very related models. And I'm going to kind of highlight their similarities and differences here really briefly on this table. And then I'm going to give a little bit more overview of the entire method before we dive into the specifics. But like I said, these are very similar methods. Just at a task level, both of them are accomplishing the same thing, generating protein backbones. RoseTTAFold or RF diffusion, sorry, uses a little bit of extra information sometimes if you'd like, but primarily they're both for protein backbone generation. They both represent proteins and residues the same way as well. So each residue is actually an element of SE3, which is a manifold that we talked about last week or 2 weeks ago. That consists of rigid transformations T that are actually composed of a rotation matrix and a translation vector. And so both of these methods use these rigid frames as a residue. And they also, because of this, both diffuse over the same manifold. So they're both diffusing over SE3, and they actually both break it down into diffusing over SO3, which is the group of rotation matrices in 3D space, as well as the translation vector. So this is just a Gaussian diffusion, and then this is going to be uniform diffusion over this. So they're both very similar in that aspect. Architecture-wise, they're slightly different, but both maintain the same property, and that main property is SE3 equivariance. Those a little bit different, but overall they just use tension and transformers to convey their model. One of the main differences, and we'll see this more as we get into the details, is the loss function. So as we kind of built up in frame diff, they kind of use a more theoretical denoising score matching loss. And this is really just trying to match the direction that you need to move in to match the data. However, RF diffusion uses more of a heuristic, and this is based on the mean squared error. And they have some justification for this, and we'll dive into that a little bit later. But the loss functions are different. So frame diff, more theoretical, RF diffusion, more heuristic based, but they both work very well. And another major difference is the pre-training. So we didn't mention this at all in frame diff because there was no pre-training, but RoseTTAFold diffusion is built on top of RoseTTAFold. And so it uses pre-trained weights from RoseTTAFold and then expands that and fine-tunes it for this actual diffusion process. Um, so let's dive into a little bit of the overview of the backbone generation process. This is a figure from the paper, but the idea is we have just a nice distribution, prior distribution for our noisy residues, and we want to denoise them into a protein structure. And this is trained by actually taking true protein structures, noising them, and then learning the reverse process. So we've talked a little bit about diffusion, so you should be pretty familiar with this. I want to mention how these frames are actually constructed because I think that's very interesting, and it's not super clear how that's done. But this is an illustration that I made earlier to kind of highlight this. So each frame T, like I mentioned, was composed of a rotation and a translation. The translation vector is really just where the C alpha is located. So if we have some sort of global coordinate frame, X, Y, and Z here, we really just have this translation vector that points to the C alpha. So it's really just as simple as that. So we have the coordinates for this translation vector. However, the rotation matrix is a little bit more complicated, and it corresponds to the orientation of this backbone. And what I mean by that is we have this global frame. If we kind of just like move that on top of this and then align it with the C alpha C bond, so you can see we just kind of rotate this with this, and you kind of have this transformation matrix here. So essentially what you're doing is lining up this C alpha to C bond with this x-axis, and then you'll have your rotation matrix. You can define this mathematically using the Gram-Schmidt orthogonalization process, using the n, C alpha, and C coordinates. But just like intuitively, you're lining up the C alpha C bond with the x-axis and forming this rotation matrix. So now we have where these residues are in 3D space based on this translation vector and this rotation. Are there any questions on kind of what these residues look like and kind of what the general shape of the proteins are in this representation?
So can we define any global friend as long as it's fixed for all the residue?
So yeah, essentially. So we're just taking a protein structure that's usually found in the PDB file, and there is some sort of like global frame already assigned to it, right? You have to have some way of telling where the coordinates are. And what the most important thing in the protein structures is really the relative position of residues. So the global frame, like where it's actually placed doesn't really matter, so much as the structure itself and the relative positions of residues. But we do have to define some sort of global frame, but it doesn't matter exactly.
Okay.
And we'll see that they kind of sidestep the translation portion by centering things later, but we'll talk about that later as well.
Thank you.
All right. So, like I mentioned, RF diffusion is built on top of RoseTTAFold. And if you don't know, RoseTTAFold is a structure prediction network. And this network came about before AlphaFold was released. We saw the results from Cast 14, and it was astounding. And some people in the Baker lab were like, hey, we can reproduce this based on just what they're talking about in their slides. And so they attempted to reconstruct AlphaFold, and their version, RoseTTAFold, does pretty well. And even their newer version, RoseTTAFold 2, is even better and is on par with AlphaFold. But it can also do multiple predictions in a single model. Anyway, for the structure prediction portion of RoseTTAFold, traditionally it takes in an input sequence. So this is just the query sequence that you're trying to fold. If I didn't mention, the structure prediction is given a protein sequence, what is the overall fold of that protein? So we're trying to predict the structure. So we give it the initial input sequence. So this is the sequence of the query protein that we're trying to fold. We also have some relatives of that protein sequence. This is usually found by forming an MSA and finding known structures of proteins that are similar based on the sequence. And then we also have some initial coordinates. The initial time through the network is actually all centered at the origin, but recycling changes this. It goes through the network and eventually you get some nice predicted structure. Going to structure generation, so this is RF diffusion, it's a little different. So like I mentioned, we're trying to generate a protein backbone. So when we have a backbone, we don't really have a sequence assigned to it. So all the sequences are actually just masked. We don't know any of the sequence. We can update this pair information here using self-conditioning. We'll talk a little bit more about that, but I'm just going to kind of leave this as a nebulous concept right now. And then this xt is the diffused coordinates. So instead of using recycled coordinates or initializing it as some other way, we actually use the previous coordinates in this diffusing trajectory. And by combining all this information, we can throw it through RoseTTAFold too, just like we do before, and get a nice predicted structure of what we think the ground truth is. And so to kind of go into RF diffusion, we kind of have to know kind of what RoseTTAFold looks like. I'm not going to go into a lot of details, but this is the main architecture. You can see the TTA stands for 3-track attention, 3-track architecture, I think, or I think the architecture. Anyway, so there's 3 tracks here. So one for the sequence. This is actually a multiple sequence track, multiple sequence alignment track, the MSA track. Then we have the pair track and the 3D track, so where the actual coordinates are being manipulated. And you see there's a lot of intercommunication between these tracks, and eventually it gets refined into the final structure. So there's a bunch of transitions. We go from MSA to an updated MSA. The MSA and pair communicate to each other. The pairs communicate and get an update from the 3D track. And then all that information for the previous 2 tracks comes and updates the 3rd track, the structure track. And then in the end, there's some refinement layers, and we have a final structure before we all would go back to the start and do some recycling. So this is the general architecture of RoseTTAFold. I don't think it's super important to go through the nitty-gritty details, but there's a bunch of attention and transformers in here. Like I mentioned, this is what the RoseTTAFold diffusion format kind of looks like. So the masked input sequence, it kind of takes this first track. It's going to go through the MSA portion. There's no other sequences because we're just doing backbone generation. This self-conditioning matrix, this nebulous thing here, goes through the 2nd layer track, and then the diffused coordinates that are actually being moved around and denoised are going through this 3rd track here. And in RF diffusion, there is no recycling. So this 6 is kind of just not present. Cool. So we kind of know what the model overview looks like. What does a single step take? So in these diffusion models, there's a lot of steps that we take in this denoising trajectory. So we start at a very noisy structure, we gradually denoise it, and then we get something nice in the end. A single step in RF diffusion looks like this. And we're going to break it down. So this first x of t is going to be your noisy coordinates at time step t. This goes into RoseTTAFold, and RoseTTAFold actually is going to predict what the denoise structures, what the ground truth structure should look like. So this is going to be x of 0 predicted, which is why there's a little hat on top. And then we're going to interpolate between these 2 steps. So we're going to take a step towards our predicted ground truth structure, is essentially what this is saying. We're going to kind of take an intermediate between these 2. And then we have our now slightly denoised coordinates here at time step t-1. There's also a self-conditioning step where they take the previously predicted ground truth structure and use that in that paired track we mentioned before. But this is how one step looks, and they threw a bunch of steps together, concatenate a bunch of these steps together, and you have your full trajectory. And so what this kind of looks like is like this. So at time step 200, so this is going to be your initial time step, we have this Gaussian distribution of translations and your uniform rotations. This goes through RoseTTAFold, and we have a ground truth prediction x of 0. This is what that looks like down here. It's still kind of a messy spaghetti monster that we can't really understand. But this is what the model thinks is kind of what the ground truth looks like. And so what we actually do to give it to the next step is do an interpolation between these. So we interpolate between these two different structures. We get something that kind of looks a little bit more like this predicted ground truth. And then this is what goes into the next step. So we throw it through RoseTTAFold again, and then we get our new ground truth prediction, which looks a little bit more like a protein, but it's still pretty spaghetti-like. We take another interpolation step. This looks a little bit more like the ground truth, and we keep going all the way down the line. And eventually what you see is these ground truth predictions look a lot more protein-like as we go. And these denoisy structures are starting to resemble the ground truth as we go all the way to time step 0, time step 1. So this is what the denoising step looks like, or trajectory looks like for RoseTTAFold diffusion. So are there any like high-level overview questions of what this denoising process trajectory looks like?
I just threw one into the chat. What happens during the interpolation step? I mean, what is the actual thing that's going on? Is like an average between the two like coordinates, xt and x0 predicted?
So we're kind of doing an interpolation on the SE3 manifold. So we're taking a step towards the correct translation vectors. So this is essentially kind of like a small step towards the ground truth coordinates, at least the C alpha coordinates. And then we also denoise the rotation matrix a little bit. So we're actually slightly making the rotation matrix of the noised coordinates line up with the predicted ground truth rotations, and then we're taking the translation vectors and trying to move those slightly to look like the ground truth predicted translation vectors. It's a little bit abstract right now, but I'm going to save some of the more like concrete details until we get into the paper, because we can actually see some formulas and things for that. But I hope that kind of like a little bit inside of that.
Yeah, thank you.
I have a question. So what I could not understand is how is RoseTTAFold taking the noised coordinates and predicting like a x hat of 0, because the architecture is supposed to take sequences. Is this one of the if you go back a slide, yeah, is this the homologous template?
So yes, kind of. So like you said, we don't have a sequence. So the sequence is really just masked out. So there's all these question marks here. That means this initial sequence is all just masked tokens going through the MSA portion at first. The self-conditioning is what goes into this pair features here. And so like I mentioned, the self-conditioning comes from these ground truth predicted structures. So what's happening is if we take this noisy structure, predict the ground truth, we then take this ground truth and calculate some extra features from it. We calculate the distogram as well as some anglegrams from this, and then use that as these pairwise template features that go through this. So that's how the self-conditioning actually works. And then the denoised coordinates, these slightly denoised coordinates, are what are going to be the input template coordinates. So we have these, initially this is going to be templates from like homologous proteins, but in our case, in RoseTTAFold diffusion, this is going to be the actual denoise or diffused coordinates. So these are our noisy coordinates that we're trying to denoise.
Yeah.
Does that help?
Thank you. Yes. Yeah.
Good. Cool. Yeah, so this is just a high-level overview of RFDiffusion. They also did some really cool stuff extending it. So the normal kind of procedure for generating structures is you kind of just sample from your priors, and then you go unconditionally all the way through the trajectory, denoising it until you have something that looks like a protein. This is the traditional way it's done, but they extended RFDiffusion to do a bunch of cool things. So the first thing that they did was symmetrize the noise. So if we take our noise distribution and actually duplicate it a couple times, so in this case we're duplicating it 3 times, and then we symmetrize it using the symmetry group C3, this is what C3 is, then we actually can have RFDiffusion produce a symmetric oligomer. So these are aligned, symmetrized around the z-axis, and RFDiffusion does a pretty good job at denoising them symmetrically as well. And we'll see kind of how they do that specifically. They also do a couple other things. So given a binding target, you can specify some hotspot residues on this target, and then it'll be able to diffuse a binder into that specific area. So if you specified maybe this helix here and this loopy region, you kind of see it get close to those regions. You can also do motif scaffolding. So if we have a functional motif that we want to preserve, so this little helix thing here, we can diffuse the rest of the scaffold. So we can hold that motif fixed, but then we can create the rest of the protein that will hold that motif. We can also do symmetric motif scaffolding. So we have this nice symmetric motif here that we want to preserve. This kind of combines the motif scaffolding and the symmetric noise to scaffold us symmetrically to generate a protein like that. We'll go into a little bit more of the specifics, hopefully, of these as we dive into the paper. Are there any like high-level overview questions of these? There are also some other extensions like potentials and things that we'll talk about later. But there are a bunch of cool things you can do with these.
Hi, Nick.
Yeah.
Yeah, I have a general question about the each step it takes from the self-conditioning. You mentioned if it's the original predicted structure look like a spaghetti, and then step by step it will looks like a real structure. But how big each step will be, and is that process controlled?
Yeah, so that is a very interesting point. And that's something I definitely glossed over because that's more in the details, and we'll see that a little bit later. But these steps are relatively small, and they kind of depend on the noise scale that the model was trained at for these, like the variance schedule that these were trained at. So we'll see this a little bit more, but the steps are relatively small. So you can see like going from here to here, it doesn't really look like this intermediate prediction here. So the steps are really small, but it's really just trying to figure out how to get it to look right. So it takes a lot of steps. See, we go from 200 to step 1 before we go here. There are ways to speed up the inference, and that involves scaling the step size, but we'll talk about that a little bit later. So I'm just going to kind of defer that to later. Sorry.
Thank you.
Cool. Any other kind of high-level overviews before we actually open up the paper? All right, let's dive in. So I'm going to switch to the supplemental materials. This is where all the meat of this method actually come in. Cool. So the first thing they do in this supplement is talk about RoseTTAFold, because this is very similar to how I set up my little overview. They talk about how RoseTTAFold is set up, represented, and how they train it. So they like I mentioned, they start with a structure of L residues, and each of them correspond with a translation vector and a rotation of that residue. And you can construct these using the Gram-Schmidt process. So this is kind of that process and the formulas and equations you need to create that. And you can reconstruct the idealized coordinates by taking some of these, the ideal coordinates based on kind of just like the statistics, and then rotate them and translate them to get your kind of your idealized coordinates in a different location. Sorry. Yeah. Sorry, I can hear you in person, but I can't hear you in the chat. But hopefully, does that help? Okay, sorry about that. Yeah, so like I mentioned, you construct the rotation matrix with the Gram-Schmidt process, and each residue has a rotation and a translation vector. I'm going to skip down here. RoseTTAFold, like I mentioned, has 3 tracks, and then some final structure refinement. It's based on attention, SE3 equivariant layers, and it updates the coordinates. We talked about this here. In addition to updating the coordinates, it also predicts some torsion angles that define where the side chain is, as well as where to place the oxygen off of that backbone. So this will actually give you all the heavy atoms of the final protein. In addition to predicting monomers, it was actually also trained for complex prediction. So they indicate some chain breaks between the different chains of your multimer using the positional encoding that's stored inside of the pair features. Here's just a giant list of all the features, but yeah, I'm not going to go through that. So all the updates are really just attention. I highlighted just kind of the major points of that. I'm not going to read through this because there's a lot, but it's really just updating the representations using attention. So MSA uses row-wise and column-wise attention. This just updates things based on an outer product. This is another axial attention, but it includes a bias term from the 3D structure. And then it uses the SE3 transformer to refine the structure. And it creates a protein graph with neighbors, and it uses pairwise features coming from the previous track and the node features from the previous track as well. It goes through this SE3 transformer to update the structure. And that's the 3rd track. And then there's the final 4 layers at the end, which are the structural refinement. These are identical to the SE3 transformer, but there's no like the other 2 tracks anymore. There's also the recycling. So previous features are recycled. So these are the pair features, the MSA features, as well as state features coming from the SE3 transformer. From the final predicted structure, we can create the histograms, and we add those as additional pair features. And then we recycle a couple times, and it's randomly sampled during training, and only the gradients are only tracked and backpropagated through the final iteration. So that's kind of like a high-level overview of RoseTTAFold. They have a bunch of losses.
Before we go on,
yeah.
The trainable weights are the trainable weights in RoseTTAFold. Is that correct?
Yes. So they start with pre-trained RoseTTAFold weights and then fine-tune on top of that.
So and then the loss propagation, they do this recycling, but it's only propagated back in the last step of the recycling? Yeah,
but the recycling is only relevant for training RoseTTAFold. In RFDiffusion, they don't do any recycling, so it only goes one time through the network.
And the loss is propagated into each time step, or
Yeah.
But they're shared weights across all the different time steps.
Right. So you have your noisy structure at some sort of time step. You pass that noisy structure and it's all its information as well as the time step, and you have some sort of time step embedding. And then you predict your ground truth structure. That only goes through the network once. All those weights are shared for every time step. So during your training process, you're randomly sampling a time step, creating a noisy structure at that time step, and then trying to denoise it. So all the weights are shared across time steps, across examples, etc.
So if we just trace all of the path that the loss is propagated through, it goes in your diagram, it's going to come out from the predictions into the loss, and then from backwards into the weights into the RFDiffusion, into the RoseTTAFold. But that backwards path, those are coming into, they're all going to be giving a shared gradient for the weights across. So it does one forward pass with lots of time steps, and then it back propagates it kind of like into all of them at once.
Yeah, so you have like a batch of examples that you're running through the network. I don't know what their batch size they actually trained with, but usually you train with a batch of multiple proteins. They're noised to a certain level, and then the gradients are accumulated across all the training examples in that batch and then updated. Yeah. Cool. So the losses that they use for RoseTTAFold aren't the same that they use for RFDiffusion, but they use similar losses to AlphaFold. For example, the FAPE loss, which AlphaFold introduced, this frame-aligned point error. They clamp it at certain distances. There's this torsion loss for the predicted torsion angles. So we predicted the side chain torsion angles as well as that oxygen, that psi. There's also losses for the bend distances and orientations. And then they also do mass language modeling and experimental prediction, some accuracy. So this is where the PLDT comes from, this prediction here. And then they do some extra predictions that are really used for the fine tuning. That was, I just sped through that, but if there are any questions, I can answer questions on that. But I sped through that because these are not the same losses that are used to train RFDiffusion. And we'll talk about that a little bit later as well.
The supplemental 3 has sort of how these are added together to form the loss table.
Yeah.
This is on page 45 of the supplement. Yeah, right there.
Yeah, so during initial training, they crop the proteins to a certain size. They have a batch size of 64, so that's what they used. And then they weight all these terms together and train like that. During fine tuning, they have a slightly larger crop size. They also include some of the more specific terms like this van der Waals term, and they train it even longer. Yeah. Like I mentioned, the data set that they use to train on includes monomers, oligomers, as well as some AlphaFold 2 structures. So this is similar to how AlphaFold used its own predictions to improve itself. They did the similar thing here where they used AlphaFold predictions to improve RoseTTAFold. Yeah, so that's an overview of RoseTTAFold. Any questions on RoseTTAFold before we dive into RFDiffusion?
How do they decide the weights on the loss function?
That's a good question. I don't know if there's like any specific way to do it besides kind of just like determining what you think is important. So for example, they have a really large weight on the FAPE loss. And so this is really constraining each frame to look like the ground truth frame. So this is very important. So they put a big weight on it, just like there's a big weight on the torsion angles. And so this is really important for predicting the side chains as well as where the oxygen is. So these that are really highly weighted are the structure prediction terms. The other terms are lowerly weighted. So this one is really trying to help the pairwise representation cover some important information like the histograms and the angles. This is kind of reasoning through the evolutionary history as well as trying to figure out which residues a protein sequence can really use in a 3D structure. This is used for the PLDT, so that was to train that head, and then this is also just, yeah. So the weights are really kind of arbitrary for the most part, at least from what I understand, and that's kind of how I do it too. But you kind of just upweight what you're most interested in and what you think is best for your model to learn that task at hand.
Okay, thank you.
All right, any other questions before we dive into the diffusion portion? All right, so let's get into diffusion. So they do a nice overview of principles of diffusion. We've talked about this previously, so I'm going to kind of go through this fast, but I'm going to slow down at the most important parts. So like we've talked about before, the forward diffusion process starts with a sample from our data distribution. So this, in our case, is a protein backbone. The data is then corrupted to produce noisy structures. So they sample a bunch of noisy structures at certain time steps based on this noisy distribution that accumulates noise from the previous noisy structures. And you noise it all the way such that the final time step at capital T is indistinguishable from a reference distribution. So this is your prior distribution. In our case, for the translations, it's a Gaussian distribution. Then this reverse transition matrices here, these kernels, are parameterized with a neural network. And to actually sample from these models, you draw a sample from the prior distribution here from capital T, and then denoise the samples iteratively. So you go from T-1 all the way back down to T until you have an example that looks like the data distribution. So the way they do this for proteins is they define the forward noising process independently over rotations and translations. So like I mentioned, our protein backbone is parameterized with the frames. Our frames are composed of a rotation and a translation. And this is very similar to how frame diff did it, but they actually do the diffusion process over the rotations independently and the translations independently. And so what we see here is this is the reverse step. So we're going from T to T-1, and they parameterize it independently or conditionally independently. So this rotation and this translation are independent of one another. The way they actually do the residue translation, forward and reverse translation or diffusion trajectories. So like I said, the C alphas are that translation vector. These are going to be essentially treated as a point cloud, and they're going to corrupt them with Gaussian noise. So they define some sort of variance schedule from the least noise to the maximum noise. And you have just from the Gaussian process, you kind of have like a forward process that's really well defined and well characterized. And you can say that the new noisy translation vector is just sampled using this from the previously less noisy sample. You noise it a little bit more, move the mean away, and then add some variance to it, and then you get something that is a little bit more noisy and looks a little bit more like the native Gaussian distribution. So you can see if this keeps increasing all the way to 1, then there will be no mean, and then this beta will converge to 1 as well, and then you'll have kind of a standard Gaussian that you'd be sampling from. To do the sampling, you don't actually have to sample from S1 all the way up to ST. So they draw these samples from the marginal distribution. So just given the true coordinates for the C alphas, we can actually sample at any noisy time step we want. And that's using this normal distribution here. And you can kind of define this using these alphas here that are related to these variance schedules here. So this is really nice and especially useful for training because given a protein sample, a specific backbone that we want to generate, we can just sample a noisy version of that backbone at any time step we want. So this makes this really efficient for training and is, yeah, we'll see this a little bit later in the algorithms. For the reverse portion, they want to create some sort of denoised structure from given a noisy structure and the ground truth. So we can do this kind of like, this is a little bit of interpolation here. So this is going to be interpolating between the ground truth translation vector here and the noisy translation vector here. So we're going to kind of interpolate them. And we can define this as a single transition using a normal distribution with this averaged kind of mean here. And so we do this to generate the denoise structure by just kind of taking a step towards the predicted ground truth, which is this Z0 hat. So this is kind of what we talked about a little bit. I just briefly glanced over with David's question earlier, where we kind of take a step towards the direction of the predicted ground truth. This is how we do that with translations. It's a little bit different for rotations, and we'll see that pretty soon. And then the variance schedule that they used for the translations is a linear variance schedule. So they go from the minimum to the maximum in a certain amount of time. And you can speed up the inference by sampling a different number of time steps. And you can just do this by actually changing what the variance schedule looks like. So the amount of noise you're adding in between steps, you kind of just scale it appropriately based on how many time steps you actually want to do. Cool. So that was very quick an overview of like how the translations work. Are there any questions about this before we dive into rotations?
So when we were originally talking about the architecture for the diffusion, one of the key innovations with the architecture was this ability to do the forward noising process efficiently. And that was, we could use the this trick to sort of add the Gaussian noises together, and that way we could have a we could sort of generate a noisy sample at any time step that we wanted. Are they using that here? And like, how does that is that, or is it not? Where does that come in?
Yeah, so that's exactly what they do here as well. So this kind of happens here. So they don't do, oh, I did not mean to do that, sorry. So instead of sampling all the way up, they do it really efficiently by just sampling, given the true ground truth structure, we can add all the variances up so that we create these alphas by concatenating, or by doing a giant product of all these variances. So this is how we do that sampling. This is that really efficient sampling. So given the ground truth structure, we can sample our noisy structure at any time step we want, and that's how we can do the training really efficiently. So yes, they definitely take advantage of that.
Were there other, I don't know how innovative their strategy of using this sort of pre-trained predictor is. Is this something that is pretty novel here, or is this an idea that they've taken from somewhere else?
I'm honestly not sure of any other method that uses a pre-trained model for this diffusion, especially protein structure prediction models for diffusion. So I think it's pretty novel. I could be wrong, but I think it is novel.
And I guess I'm curious on how different it is. Like, for instance, if you take the model that's trained for RF diffusion and just do structure prediction with it, like in the sense of using those weights back in like RoseTTAFold, does it work, or is it like a completely different? Is it learning something that's not like, you know, how you, I know you can do the ablation or the fine tuning or not, but it gets kind of tricky in terms of the, like, you know, initial conditions and all this, but like, you know, how good is it in terms of RoseTTAFold itself?
That's a very interesting question. I don't know. My kind of intuition is that you would kind of get garbage out of the network, just because they were fine-tuned and trained for specific purposes. So I don't think if you add sequence information and template information to a RoseTTAFold diffusion trained model, I don't think that would be very good at predicting the structure of that protein. But that is something we could totally do. Like, we have the weights for each of these networks, and so we could run an example through. That would be a very interesting test. I don't know if anybody's done that. Cool. Anything?
Hey,
any
Nick,
other questions? Yeah.
when you train RF diffusion during the training, do you have the sequence information?
No, not natively. We'll see that a little bit later they do condition on some information. So if you're doing like the motif scaffolding problem, for example, some of the side chains of the motif or the sequence information of that motif that you want to scaffold could be important for generating the protein. So it is possible to inject that information in, because there's also that sequence track in RoseTTAFold. So that's not something that they do for the unconditional generation, but for like the motifs and for the active site generation, that is definitely possible.
So how does it, I mean, this gets back to a question that was asked a little while ago. How, if there's no sequence information during training, how can you expect the model to I mean, because the loss function is to the ground truth native structure, right? How can you expect the model to predict the native structure without a sequence?
So we're only working with backbones, which is how we do that. So because we're just using the backbones from the PDB, essentially, we're just stripping all the side chains away, all the sequence information away, and trying to predict the distribution of backbones. So that's how they get around that.
But I guess also maybe is it that like from the really noise structures, you don't really expect it to predict the native structure. And it's probably only the ones that are noise just a little that actually
Yeah, exactly.
get closer to the.
So I'm going to jump back here real quick, just because I think this kind of highlights that. If you look at these predictions down here, only until you're very close to the final time step do they actually look protein-like or resemble the final predicted structure. So
it's not even that whether they look protein-like. They have to have a low loss. They have to look like the natives, whatever native structures used for this training, right?
Yeah.
Yeah. All right. Thanks.
Mm-hmm. Okay, let's go ahead and push forward into the rotations, because this is where it gets a little bit more complicated. And we saw a little bit this for frame diff, but so the rotations live on SO3, which is this manifold of all rotation matrices in 3D. And you have to define some sort of inner product on this in order to do the Brownian motion on this manifold. So we do it on the tangent space of the manifold. So they define this inner product here. This is just really the canonical inner product on SO3, but it's a scaling of the Frobenius norm. And we'll kind of see that a little bit later. This Frobenius norm actually is used in the loss function. But yeah, we'll see that a little bit later. So they have this inner product, which defines how we can move around on this tangent space. And so I'm going to try to give a little bit intuition on what I think about the tangent space. I don't know how well this is going to go. No, that's not what I want. But I like to think of the tangent or the manifold of SO3 as a ball. And you can do that if you like interpret the I'm really struggling trying to find how to draw. I think I'm just gonna skip because I don't know. Oh, I can probably use Zoom to draw. Yes, okay. I'm gonna use Zoom to draw. So we have this sphere in 3D. So this is gonna represent our SO3 manifold, and you can do this if you use a different representation for the rotation matrices. Because we can represent rotation matrices in the Euler axis, like the axis angle representation, we can define, if this is the origin, we can define a vector going to the surface of the sphere, and that can kind of represent the axis of rotation around which we're rotating. And so this represents a point on the sphere, and if we just travel around on the sphere, we can get a bunch of different angles or axes we're rotating around. We can define the tangent space, which is really just a kind of a generalization of the tangent line. For this manifold, it's going to kind of look like a plane. So we'll have like a piece of cardboard laying on top of our ball here, and this is going to be our tangent space at this point here. And we need to define some way to move around on this tangent space. So this is where the Brownian motion comes from. And the way that they really do this is they parameterize this Brownian motion. Sorry, it's going to get overlaid. Let me, I'll delete that. But they are able to do this Brownian motion over this tangent space using this isogaussian SO3 distribution here. And it's parameterized by the initial rotation and a time step t. And you can really get a really nice uniform distribution on SO3 by sampling from this. So there's this complicated formula with this infinite power series here, but I'm just going to kind of gloss over that because this is used in the score approximation, which we'll see later, but this needs to be truncated because it's infinite, but they use this to help compute where you need to move in the tangent space. So we are on this cardboard piece, we move in a direction on that cardboard, and then once we've moved in that direction, sorry, I'm going to draw again. So, oh no. Okay. So we have our, okay. We have our ball, our SO3, we have our tangent space at some position here. Once we take a step in the direction that we need to go on this tangent space, we are now no longer on the ball, which is a problem. We need to remain on this manifold, right? If we want it to be like a realistic rotation matrix. So the way they do this is with a geodesic random walk. So once we have this direction we need to go, we're able to bend it back and put it back on the manifold. So now we'll be back over here in this direction now. So we take a small step in the tangent space. We leave the manifold, and then we kind of bring it back to the manifold by bending this. And now we kind of have a curved direction, at least on this manifold. It kind of looks curved because we're on the sphere. That was a little abstract, but hopefully that was somewhat clear. But yeah, so we have this isogaussian. We can do the forward motion by sampling from this isogaussian SO3 distribution, the marginal distribution. And this is, I think, a really important point here. So the translations, like I mentioned, converge to the Gaussian distribution as the time step increases. The rotations, however, converge to a uniform distribution on SO3. So we have a distribution on that ball. Now in the final time step, capital T, it's completely uniform on the ball. So any rotation axis is as equally likely to be sampled as any other, which is a little different from this Gaussian distribution, right? Because this would be sampling around 0 a lot more than anywhere else. So like I mentioned, the reverse step for the updates for the rotation, you have to take a noisy step in the SO3, the tangent space. So we take a step on the cardboard, but then we left the ball, so we have to project back onto the ball. So we project back to the SO3 manifold using this exponential map. And that's really comes in later. So I mentioned that there's this geodesic random walk, which is how we map from the tangent space back to the SO3 manifold. So this is the most important quantity here. Really, this is the direction we need to move in the tangent space. So in our case, this is how we go from the noisy rotation to something that looks more like a data rotation. And then this term here is really just adding some extra noise to that. So this is kind of handling the Brownian motion portion of that. Ooh, I messed something up. I didn't know that was possible. So this is kind of the Brownian motion portion of it. And we do this exponential map to get back to the rotation. So yeah, this is the Stein score, the score matching portion of score matching. And then we have an exponential map from the tangent space, so that piece of cardboard back to the ball. And yeah, so this is kind of how we define the forward and reverse trajectory for rotations now. And this uses a different schedule. This uses a quadratic schedule rather than just a linear one. But once again, that's just a hyperparameter. So learning this actual reverse trajectory is possible using score matching, and this is done in frame diff. This is like that very theoretical way to do it. But they wanted to do an approximation that really leverages the ability to predict the denoise structures. So RoseTTAFold is really good at predicting structures. So can we use that ability to advance our diffusion? So the way they do that is they form this approximation here. So the score that we're really interested in is the score given the score of going from the wild type ground truth structure to the noisy structure. So this is the direction we want to move to get to the ground truth structure. We're only really interested in the rotations, at least for this portion. So we're going to just use the rotations here. We can approximate this by using the ground truth rotation. So this is the predicted ground truth rotation, sorry. So this is what is coming out of RoseTTAFold. And we can approximate the score using this. And this is the same thing as using the isogaussian, using the predicted rotation from RoseTTAFold. So this is kind of how we leverage RoseTTAFold's ability to predict ground truth structures in a way to approximate the score that we can use for the denoising trajectory. So once we have this approximation here, we can throw it into this equation, or we actually can write out this equation to use that from that power theories that we used above to compute the direction we need to actually move in. Yeah, that was a very like abstract. So yeah. And the way to actually do the rotations in the reverse process is you take the step in the direction, and then you really just rotate it. You rotate what you were previously at by this extra rotation, this step you're making, and then you'll get your slightly denoised structure. And so, yeah, we can see this approximation here. They approximate the power series, and then they can approximate the score, the direction, the step we need to move by taking the predicted rotation and computing that score approximation using all these big equations. They do some proofs to help justify it. Yeah, so that's kind of a high-level overview of the rotation process of forward and reverse diffusion. Are there any questions on this?
So is the main difference between RF diffusion and friend diff is that RF diffusion uses predicted structure from RoseTTAFold as the ground truth structure when calculating that loss, or
Yeah.
there? Okay. And in
Yeah,
friend diff they just use the ground truth, right?
Yeah, so they so in RoseTTAFold they use or RF diffusion, they predict the ground truth structure and then back calculate what the kind of noisy interpolation to denoise it is. The other main difference I would say is the loss function. So RF diffusion doesn't actually use the denoising score loss. It actually uses a MSC loss, which is kind of just a heuristic approximation of the denoising trajectory. So this is kind of where this comes in here, this mean squared error loss. So they actually do this mean squared error loss on the translations here and the rotations here. And this is where the main difference, I think, comes from, besides, using a pre-trained structure prediction network for diffusion. This is where the other major difference comes from, is besides also like architecture. But this loss function is not really like super grounded in theory. It does have some like justification that they explain a little bit down here. So you can kind of, when it's Gaussian, you can kind of do a rescaling of this KL divergence using this expected error of the prediction and the noisy observations. So this is really easy to do. This is kind of just approximating what the noise was added, or the score that you need to move in. And this really works. This is well defined and is justified when you're using Gaussian noise. But for rotations, this isn't entirely justified. It likely doesn't hold for iso-Gaussian SO3, but it's pretty sensible to use, and they justify it down here. So this loss function is also really how it differentiates itself. I hope that helped a little bit. And also, I know we're out of time. So yeah, feel free to head out if need be. If you want to stick around, I could talk more. But
well, thanks, Nick, for doing such a nice detailed overview of it. It's amazingly productive. Like the RF diffusion works so well in terms of their benchmarking, and also I know that a number of people in the community have applied it to their own specific tasks. And it's hard to know exactly what the secret sauce is. And it would be really fun to sort of explore related architectures. Like we did frame diff, but there's other ones coming out as well, to try to figure out what pieces are, what's the magic sauce that makes this particularly work. So I think that will be what we'll sort of explore more, and as we go forwards, is it the pre-training? Is it the diffusion? Is it the sort of this combination of these different losses? And is it the data? What aspects are really, really essential for it? So I know we're out of time, but I think this reading group has been fun. I know I've certainly learned a lot from it. So, if people have other feedback in terms of structure or other ways that we have it organized, definitely let us know and we can adapt. But otherwise, we hope to see you in 2 weeks, and otherwise be safe and study hard.
Thanks, everyone.
Thank you.